So I am Kathryn, as Nikolai has introduced. Thank you very much to him and the organisers of this meeting for giving me the opportunity to talk about what my lab does. I am very surprised, I think, to end up speaking at a single-cell proteomics meeting because, as Nikolai has alluded, I've never worked with a single cell. And in fact, the experiments that I'm going to describe to you, the minimum number of cells that we've ever started with is 10 million. So this is not a single cell talk. But I hope that some of the messages that are coming out of the research that we're doing in my lab will resonate with the single cell proteomics community. So we are interested in the subcellular proteome. We all know that proteins must traffic to the correct location to carry out their function. And we also know that it's becoming more and more apparent that when proteins get this wrong and a protein ends up in the wrong place, then this can be the underlying factors for many disease pathologies. So we need to be able to map locations of proteins in cells and also in disease states where this starts to go badly wrong. So we also know what controls, to some extent, where a protein is going to end up in a cell. It can be its sequence. So some proteins will have signal sequences that direct them to different subcellular niches. It could be through post-transcriptional and post-translational modifications. Different splice variants of proteins may end up in different places. If a protein is phosphorylated, it may end up in a different place to when it isn't phosphorylated. Also, location will be dictated to some extent by interactions. So these could be interactions of proteins with other proteins, or with RNA, or with other biomolecules. And it's also dictated by some extent as well to where a protein is synthesized. So some proteins are synthesized in situ so that they can immediately start doing their jobs in the right location. Also, and I'm not going to talk about this at all today, it can be to do with conformation. So many proteins are shapeshifters, and depending on which shape they adopt, may dictate their subcellular location. So of the other 4 points here, I'm going to talk in turn about methods that we've developed or are developing in my lab to enable us to determine the functional aspects of protein subcellular location. And why this is important, of course, is that proteins are not static individuals. They're very dynamic. And depending on a cellular perturbation, such as stress or nutrient status, or even circadian rhythm, what time of day it is, this may lead to subcellular relocalization, with proteins either being involved in other jobs or in other complexes at different times or in response to different perturbation. So we can achieve a map of the intracellular proteome. There are lots and lots of different complementary approaches which will tell us where proteins are in cells. But we need to make functional sense of this. We need to work out why proteins are localizing and what is the functional consequence of this relocalization. So earlier this year, myself and leaders in the field within spatial proteomics put together a very nice summary document for Nature Reviews Methods Primers on the different approaches to look at the subcellular proteome. So I direct you to this to get more details, because I'm only really going to scratch the surface of the different methods that are available. But by way of a little bit of background to what these approaches are, there are many of them, and which one we choose to take is going to be dictated by the question we have. So it may well be that we have a protein of interest, we know what it is, we want to know where it is, and so we're going to use some sort of imaging method. We're either going to tag it with a fluorescent marker protein or use immunocytochemistry methods. So these are very, very powerful methods, and are going to be taught a lot about later on in this conference. Maybe our question's a little different. Now what we want to know is what components make up our subcellular niche of interest. So maybe we can purify this or greatly enrich whatever this niche is from cells, or we can label our niche of interest. And a very good way of doing this is to use the lovely proximity tagging methods, APEX or BioID, where we'll biotinylate a particular subcellular niche, pull that niche out with streptavidin, and then by mass spectrometry, typically, we would identify the components. And there's beautiful work coming out of Alice Ting's group and that of Anne-Claude Gingras, who have done this on approximating a cell-wide scale to look at different subcellular location. Now, if you do these methods singularly, what these methods are not very good at is telling you if you've got proteins that are present in more than one location. So to be able to get a map that will give this type of information, then there are cell-wide methods. And these methods are largely based on protein correlation profiling, and I'll talk a lot more about these methods in a moment. There are also various other approaches that we could take using mass spectrometry, either mass spec imaging or imaging mass cytometry. And it's also important, I think, to put all these different approaches in some sort of context. So for the most exquisite subcellular resolution, then the imaging methods are always going to be the ones of choice. However, in terms of proteome coverage, we're going from a single protein per experiment up here to tens of thousands of proteins with the methods towards the bottom of this slide. Some methods are sympathetic to proteins that exist in multiple locations, as I've said. Some methods are better at giving cell-to-cell heterogeneity, which are the imaging methods. And the other methods, which I have down here, what we have to remember all the time is because we're using multiple cells in our experiment, we are returning information on the cell average, so the average location of a protein across millions of different cells. So I want to talk about the cell-wide methods. So these are the correlation profiling methods. The ethos behind them is not new. It was first published 60-odd years ago by Christian de Duve, and he argued that it's actually remarkably difficult to get a pure fraction of any particular organelle. So why bother? Why not just carry out some sort of cellular fractionation in which you partition the subcellular components in different fractions and then look at the protein abundances across those fractions and work out what is a typical pattern or a typical correlation profile for marker proteins for a particular organelle. So let's imagine we've done this, and for our red organelle, so this would be the lysosome, it's peaking in fraction number 1, the Golgi apparatus is peaking in fraction number 2, and so on and so forth. Then if we have a correlation profile of a protein of an unknown location, we can look at its abundance across our fractions and then match it by some method to see what it most resembles. So in the case of this one, it's most like the endoplasmic reticulum, but it may also be that a protein is in multiple places, and then we'd have some sort of composite of these correlation profiles. So the second unknown perhaps is partly in the endoplasmic reticulum, but also partly in the mitochondria. So this is the basis of correlation profiling. It's been published now in many different guises. We got into this methodology very, very early on, producing a paper an embarrassingly large number of years ago, where we talked about the method that we developed, which we call LOPIT. So this is our version of correlation profiling, and that stands for localization of organelle proteins by isotope tagging. We have two different versions of it. We have hyperLOPIT, which is based on separating out organelles by their density using equilibrium density centrifugation. And we also have a quicker method, which is based on differential centrifugation. And I've written here the number of cells we would typically start with for both these methods if we were working, for example, with cell culture. So these are nowhere near single cell methods. So to give you some idea of how we go about this, the first thing you need to do is to lyse your cells, whatever they are, using a method which maintains organelle integrity. So you can use ball-bearing homogenizers, Downs homogenizers, and then we have our different approaches of fractionating out our organelles based on either differential centrifugation or density equilibrium centrifugation. So then what we do, once we've got our fractions, it doesn't matter quite how we've got them, we could use detergent solubilization, any method which would give us some sort of fractionation of our organelles. We then, in my lab, use TMT, so tandem mass tags, to work out the abundance of proteins across our fractions and be able to deliver a correlation profile for every protein in our experiment. So this is the sort of data that we would get. This is a PCA plot, a PCA projection of data. This is fairly old data now from a mouse embryonic stem cell. Every spot is a protein. If it's coloured in, that means it's been assigned to a particular subcellular location, or it's a marker protein. So it's very important to have marker proteins to be able to distinguish what the different clusters mean. So if it's coloured in, it's been assigned to something we know. If it isn't, then we class these proteins as unknown, and they can be unknown for a variety of reasons. One is that they cluster with some subcellular species for which we have no markers, so we don't know what it is, or they are proteins that are in more than one place. So what these types of plots give you is the steady state location of a protein. So if part of it is in the nucleus and part of it is in the cytosol, then it's going to end up somewhere down here. But using this method, we can look at discrete locations and now are readily returning thousands and thousands of proteins per experiment. So this is a cell-wide method. Importantly, what we see in just about every sample that we've ever used with LOPIT is that up to 50% of the proteome can be in this intermediate position, so of no fixed abode. So that's an important fact to remember. So then we can do all sorts of interesting things with our data. We can look to see if we've got different isoforms and if they partition to different parts of the cell. So the example I'm giving you here is leucine aminopeptidase 3. There's a long version, which has a mitochondrial import sequence at the N-terminus, and a shorter version. And we can see that they end up in different parts of the cell. And interestingly, if you look to see what LAP3 interacts with, according to string, then actually its interaction partners are also spatially restricted. So it's very attractive to assume that the long form interacts with its partners in the mitochondria, where the short form is in more than one place, and it can interact with these partners in the cytosol and these partners in the secretory pathway. But we'll have very nice resolution by this method, including sub-organelle resolution, because what we have here in pink is nuclear chromatin, where in pale green we have nucleoplasm. And we can also see large protein complexes, so we have the two ribosomal subunits here and the proteasome down here, and we've got nicely resolved components of the secretory pathway. We can also start to then map on other features of the proteome. So it's interesting to see where members of protein complexes live. And it's also reassuring to see that these complexes stay together in our methodologies. And we can start to look at very defining features of the complexes. If you look at this one here, which is the red open circles, you can see most of them are part of the plasma membrane. But we have a rogue component over here. So this might represent a transient interactor, or it might represent the fact that this component, whatever it may be, is involved in more than one complex, in more than one part of the cell. And again, we're seeing the average. We're seeing the steady state location of the components of these complexes. So to be able to make sense of our data, we've needed to develop a whole host of different analytical pipelines. And what we did some while ago now, and this was the brainchild of Laurent Gatto when he was a postdoc in my lab, is a complete R-based infrastructure which allows us to take in our data to be able to manipulate it, to classify proteins, but also then to be able to visualize it using many different types of projections. So I showed you PCA analysis, but we could use all sorts of different projections if we wanted to. I'm not entirely sure this video is going to play, but more recently, Lisa Breckles, who's a postdoc in my lab, has produced a series of different graphical user interfaces associated with our data sets so that you can go into our data and you can have a look where your favorite proteins are, what also is clustering around them in our data. And this is fully interactive, fully searchable. So if I can move on, the other thing we've had to do is to develop methods to look at the probabilities of proteins being in particular locations. So these are packages which have been continually added to ProLoc. And the one I want to talk about here is TAGM. So this was developed largely by Oliver Crook, who was a PhD student in my lab. And what he did was to apply Bayesian theory to be able to create probabilistic models of our data. So we want to know what is the probability of a protein assigning to a certain subcellular location, but we also want to know uncertainty. And by that, what I mean is what the chances are of this protein being in multiple different places. So what this probabilistic model allows us to do is to characterize the uncertainty in location. And therefore, this is very sympathetic to the great number of proteins that are in more than one place. So if we look at the example down here, exportin 5's job is to carry cargo to and from the nucleus to the cytosol. So you would expect it to be in more than one place, and we can get the probabilities of these locations from creating this probabilistic model. What Ollie's gone on to do then is to also use this method in a semi-supervised way, such that we can find clusters of proteins that are behaving in the same way, where they've not necessarily been annotated. So this would be very good, and has been very good, at finding novel subcellular niches that proteins are clustering to in different cells. So the applications of LOPIT, we can use this two ways. We can either produce a static map, or we can produce a moving map, so dynamics of protein relocalization upon some perturbation. And I'm going to give you an example of both. So the first I want to give you is a very nice story that has come out of a collaboration between myself and my colleague Ross Waller in the biochemistry department in Cambridge. And Ross is interested in this beastie up here, which is Toxoplasma gondii. It is a single cell obligate parasite. It's a member of the Apicomplexa family. It is very prevalent in the human population. Its intermediate host is the cat, and all of you who are cat owners are likely to have been infected with Toxoplasma. It's not going to do you very much harm unless you become immune suppressed. So it is a pretty unpleasant, or can be a pretty unpleasant human parasite, and we don't know a huge amount about, or at least we don't know a huge amount about its proteome. It has a great variety of different subcellular species and organelles that have evolved over the years to allow it to become an obligate parasite and to infect the host cells. So we applied the hyperLOPIT protocol, and this was the work of a joint postdoc, Konstantin Baryliuk. What he did was, using nitrogen cavitation, was to be able to break open these Toxoplasma, which are quite tough, they've got crispy coats, but he did it in such a way to preserve the organella integrity, and then we got this very beautiful map here. So this is a t-SNE plot this time, and you can see these beautiful clusters which are clustering with markers to a lot of these very specialized organelles. So what we were able to do was to return subcellular location for 65% of the proteome, in 26 different subcellular compartments, many of these being newly assigned to these compartments, and many which went on to be verified by immune microscopy. For the vast majority of these proteins, there is no documented function. And what is quite cool is that you can then go on and have a look at the evolutionary trajectory of orthologs from these proteins within Apicomplexa, and see which organelles have come into being during the evolution, because we now know the sets of proteins that belong to these different organelles. So you can see what came into being as the parasite went from being a free-living organism to a parasite, how it's adapted to its host, and what are the sort of bare essential organelles that have persisted through evolution. So I'll move on now to how we can use the spatial proteomics methods to look at subcellular relocalization upon perturbation, so dynamic maps of the cells. Now, we've got to think about this very carefully. We're going to end up with multiple sets of data, and from that, we want to be able to map proteins moving from A to B, proteins moving from mixed localization to a single localization, and vice versa, and also proteins that don't move at all. So we want to be able to particularly look at proteins that are in an intermediate location and see whether they are moving to somewhere of a more fixed abode. So to be able to do that, again, we've had to return to Bayesian statistics and to create bespoke analytical pipelines so that we're getting true probabilities. So we want a probability of a protein moving now from A to B, and also we want to be able to map this uncertainty so that we can look at proteins that are in multiple places and how they are changing or how their distribution is changing upon perturbation. So again, this is work by Ollie Crook in collaboration with Paul Kirk at the MRC Biostatistics Unit in Cambridge, and the package that he's come up with is Bandle. And what Bandle does is to compute this probability of a protein being differentially localized upon perturbation and quantifying the uncertainty associated with that. It's a very flexible approach. We've applied it to not only data from my lab, but from similar data sets from quite a few other people's laboratories. We've also applied it to data that we have created in silico and shown that it's a very high functioning. It can be used in a very flexible way, irrespective of the N number, and also in comparison to some other methods that are out there at the moment. It's very sensitive, finds additional relocalizations, and has reduced type 1 and type 2 errors. So to talk about one of the studies in my lab where we've looked at protein relocalization upon some sort of perturbation, I want to turn to the innate immune response. So what we've done here is to take a monocytic cell line and to treat it with LPS, a lipopolysaccharide, which is present on the gram-negative bacteria, which elicits and promotes the innate immune response. What happens to these monocytic cells is that they start to differentiate become more like activated M1 macrophages. So the aim of the experiment was to capture the global changes in the proteome on a cell-wide scale upon activation of a pro-inflammatory phenotype. But there was a slightly more fundamental question underlying this, which was if you carry out an experiment where you have applied some sort of perturbation and then measured the proteome, what the vast majority of people do is to lyse the cells, and then they pin all their analysis and all their conclusions on changes in protein abundance. But it may well be that there is no change in protein abundance and that the proteins are just shunted from one part of the cell to another part of the cell in response to this perturbation. And without carrying out some sort of spatial pipeline, all this relocalization information goes out the window. We have a wonderful saying in the UK, which is throwing the baby out with the bathwater. So this may well be what is happening in a lot of different quantitative proteomics experiments where spatial information is not captured and only abundance information is. So we thought we'd see what the extent of this is in our system. So this has been a lot of work by a former postdoc in the lab, Claire Mulvey, aided and abetted by Lisa Breckles. So what Claire did was to firstly do a time course. So she got all these abundant measurements in her cells across a 24-hour period using a very standard quantitative proteomics pipeline. She also then chose two time points, 0 and 12 hours, and I'll explain in a moment why she chose that time point, and carried out hyperLOPIT. So she's ended up with two sets of information, how proteins are changing their abundance in time and how proteins are relocalizing. Now, the reason she chose 12 hours is that she showed that there were major proteomics changes at this time point, but the cells were retaining a non-adherent phenotype. So she's wanting to capture the early response of the system without having a very different phenotype of the cell. So what she showed was that between her two time points, there was quite a lot of subcellular relocalization. So 112 proteins are moved from one place to another. Some had mixed localization and then at 12 hours had moved lock, stock and barrel to one particular location, the reverse, and proteins in mixed locations that moved to different mixed locations. Of great importance, only 2 of these proteins showed any change in abundance. So the rest were moving with no concomitant change in their abundance. And Lisa's produced this beautiful waterfall plot to see where the majority of the movers were going to and from, and it showed that of the movers, a lot of them were involved in nuclear to cytoplasmic shuttling, plasma membrane and cytoskeletal modelling, and the formation of the autophagosome. So to just give you it's a very rich data set, but just to pull out a couple of points from it, she showed a family of Rho GTPases. So they were cytosolic at 0, but after 12 hours in LPS, they'd moved lock, stock and barrel into the plasma membrane. This was, in some cases, not a shock, because in the case of one of the components, CDC42, this had already been shown using microscopy methods. But again, importantly, this protein is not changing in abundance during this process. It's changing solely in location. Of the 2 proteins that did change in both abundance and location, the first one, SQSTM1, is part of the autophagy receptor, and it's increased in abundance over this time period and has moved with other receptor components as a unit from the cytosol into the plasma membrane. The other protein, CLEC11A, is a cytokine, and this showed a decrease in abundance with time. This has moved from the lysosome into the endoplasmic reticulum, and what we're thinking is happening here is that at the 12-hour time point, it's being secreted. So it looks as if there is a decrease in protein abundance because it's being secreted, and what we're seeing is the ER component of this protein that's going through the secretory pathway. So just to show that from these data, you can start to pick up some very nice biological features to go off and investigate by other methods. Also, because Claire had this temporal data, she could show clusters of proteins that temporally are behaving in the same way, also tend to co-locate. So that is the method, and this is how we can use it to gain static maps and also dynamic maps of the proteome. But we want to know the functional significance and why this is happening. Why are proteins moving from A to B? So one of the things that we are looking for is to see what the influence on post-translational modification is on protein localization. So I'm just going to show you a static map of this. But what we want to know is if a protein is phosphorylated, is it in a different place to the unphosphorylated version of the same protein? So to be able to do this, you need to get a map of the total proteome, and you need to be able to get a subcellular map of the phosphoproteome, or whatever particular post-translational modification you're interested in. And to our minds, it's very important to do the following, which is to look at the steady state location of the protein, or peptides from that particular protein, and how this changes on a peptide scale if you modify the peptide. So maybe that our protein down here, here's its correlation profile, perhaps it's in 2 different places. The phosphoproteome, or the phosphopeptide, might have a different correlation profile. And what we think is important to do is to also look at the correlation profile of the unmodified peptide. So now what we're saying is that if the proteins are modified at this particular location, then it's in this place. If it's modified, then it's somewhere else. So we've tried to map the correlation profiles of matching phosphopeptides and their unmodified versions. So this is the work of Anya Andreeva in the lab. And what she did was to carry out the LOPIT procedure. In this case, it was LOPIT DC, so it's the differential centrifugation version, again using TMT. When she's got her TMT labeled peptides, then she can take some of it off to get a map of the total proteome. and the rest she can use for enrichment of particular post-translational modifications. And she's done this sequentially, fishing out the phosphoproteome, but also various other post-translational modifications, which I'll talk about in a moment. When we project the correlation profiles of the phosphopeptides onto the total map, there's an awful lot of phosphopeptides that map onto both the nucleus and the cytosol, but they're generally in lots of different parts of the cell. And of interest, she was able to find matching pairs, so over 500 matching pairs of phosphorylated proteins and their non-phosphorylated equivalents. Then she could look at their correlation profiles to see how they changed. 72 of them actually showed a difference. The rest didn't. It didn't matter whether the protein was phosphorylated at that position or not. It wasn't moving. But of the ones that she did, I just want to pull out one particular example, and this is from the protein EIF4E binding protein 1. And what she showed is that the correlation of the phosphopeptide, which corresponds to a doubly phosphorylated 2-threonines version of the peptide, was cytosolic, but the unphosphorylated version of the same peptide had a correlation profile which mapped better to large protein complexes. Now, given what this protein does, this sort of makes a lot of sense. What this protein does is to bind to EIF4E, and prevent it from being recruited to initiation complex to bring about the start of translation of an mRNA by helping recruit the 40S ribosomal subunit. So if it isn't phosphorylated, that's what it's bound to. When it gets itself phosphorylated, then it detaches from EIF4E and off it can go and initiate translation. So when it isn't phosphorylated, it's part of a protein complex. When it is phosphorylated, then it's free to go off and do its own thing. So that matches nicely what we're seeing. Of importance, I think, is to also think about the fact that a protein can be modified in many different ways. So what Anya did was to also measure the succinylated proteome by enriching succinylated peptides using an antibody. And I just want to point out this protein. So this is a dead box helicase. It's DDX21. Its steady state location is in the nucleus, and its job is to sense pol 1 and pol 2 transcription, RNA polymerase 1 and 2 transcription in the nucleus. When it's phosphorylated, it appears to be cytosolic. When it's succinylated at a particular position, it appears to be mitochondrial. Now, if you look at the UniProt entry for this particular protein, it's in all three places. So what we're showing here is, depending on what types of modification the protein picks up, will dictate its subcellular location. Now, I'm not saying it traffics to these places because of this modification. May well be it traffics for some other reason, and then is succinylated in the mitochondria and is phosphorylated once it gets to the cytosol. That we can't say exactly why the protein is moving. We can just see what the consequences of that move may be. So just quickly, I thought I'd talk about interactions. So my lab over the last few years have been very interested in the RNA binding proteome. And we, along with others, including our next speaker, Jeroen Kruisveld, have come up with quick, easy and very sensitive methods to fish out the RNA binding proteome. Our version of this method is called OOPS, or orthogonal organic phase separation, and it works in the following way. If you cross-link RNA to protein and then carry out an aqueous organic partitioning, free RNA goes into the aqueous phase, free protein goes into the organic phase, and anything that's cross-linked ends up at the interface. So if you isolate the interface, wash it a few times, you can release the RNA binding proteome by using RNAs, or you can release the RNA that's been bound to your proteins by treating with a protease. So we reasoned that we could use this method subcellularly to look at the location of RNA binding proteins in the cell, and importantly, where they are doing the job. So do they bind RNA in a different place to the bulk version of that particular protein, so when it's not binding RNA, for example. So what we did, and I won't have not time to go into this, is very much change the way in which we carry out density centrifugation and differential centrifugation methods to make them RNA friendly, and then carried out OOPs on each of our subcellular fractions. And then we were able to see the following things. So on the right-hand side, I've got the maps of the free protein. On the left-hand side, I've got the maps of the RNA binding proteome. So in the case of various components of the translation machinery, if they're free and not binding RNA, they're cytosolic. If they are binding RNA, then they're part of ribonuclear complexes. You can also see examples of where it makes no difference whatsoever to the location of a protein if it's binding RNA or if it isn't. So in the case of tRNA ligases, they're cytosolic whether they're binding RNA or not. Just to draw your attention to what we have in blue here, which is the ER. So here's the ER for the free proteins. In the case of the RNA binding proteome, there seems to be two separate clusters. And when we look at those clusters individually, cluster number 1, the RNA bound protein is ER and the free protein is also ER. But in cluster number 2, this is not the case. If they are binding RNA, they're ER. If they're not binding RNA, then they're not, their steady state location is not endoplasmic reticulum. And the set of proteins threw up some surprises. They're enriched in limb domain proteins and cytoskeletal proteins. And certainly this requires a lot more investigation. So my last slide, this is just a teaser really, that we've now redesigned the way that we can do LOPIT so we can fish out the transcriptome of our fractions as well. So now what we can get is matched maps of both the transcriptome and the proteome. We've called the RNA version of this method, we've called the RNA version of this method LORNA, or the localization of RNA. So we're busy using this, the LOPIT-LORNA combination to help us to work out the relationship between a protein and its corresponding transcript, and to try and identify, particularly upon perturbation, where we've got potentially changes in localized translation. So, in summary, I hope I've shown you, even though we use millions of cells, that mapping the subcellular location and how it changes gives insight into function. I also hope that I persuaded you that half the proteome exists in more than one place. We can map post-translational related localization on a cell-wide scale, and also the RNA binding function of the proteome can be spatially restricted. And also I've given you a bit of a teaser that we can create matching map now of RNA and protein. And we hope that these methods will give essential insight into mislocalization and its source, aberrant translation or aberrant trafficking. So with that, I have a great many people to thank, both past and present members of my lab, lots of collaborators, all our funding sources, and I'll leave you with an advert if your appetites have been whetted for more information about the spatial proteomics approaches. This is a workshop that's happening next week, which is organised by Emma and myself through a European Infrastructure Consortium, EPIC XS. Thank you very much for your attention.